Gluconeogenesis by Monique Alamán and Sanaz Khamisi. Overview of gluconeogenesis. This is an anabolic pathway, which is the synthesis of glucose from non-carbohydrate sources. It is the basic reversal of glycolysis with three exceptions. The first one being pyruvate kinase is replaced by pyruvate carboxylase and phosphoenolpyruvate carboxykinase, also known as PepCK. PFK1 is replaced by fructose 1,6-bisphosphatase, and hexokinase is replaced by glucose 6-phosphatase. This is a ubiquitous pathway observed in animals, plants, and microorganisms. In animals, it occurs mainly in the liver and kidneys. In plants, it is seen during seed germination and post-germinative growth. Multiple cellular components are involved, including mitochondrial, cytosolic, and the endoplasmic reticulum. There are 11 reactions in this pathway, and we will discuss them step by step. Gluconeogenesis begins with pyruvate. The first step is the carboxylation of pyruvate to form oxaloacetate. This reaction is catalyzed by pyruvate kinase and requires ATP. This occurs in the mitochondria, as you can see in the picture to your right. Pyruvate kinase is activated allosterically by high concentrations of acetyl-CoA. It carries a biotin prosthetic group which functions as a carrier of carbon dioxide. When activity of the TCA cycle is low, the oxaloacetate produced enters the gluconeogenic pathway. In the second step, oxaloacetate is decarboxylated and phosphorylated to produce phosphoenolpyruvate. This is accompanied by the hydrolysis of GTP to GDP. This reaction is catalyzed by the enzyme phosphoenolpyruvate carboxykinase, PepCK. In some species, it occurs in the mitochondria. In others, it is cytosolic, and in some species, it occurs in both locations. Due to lack of specific transporter proteins, oxaloacetate cannot be transported across the mitochondria. So, in species which only have cytosolic PepCK, oxaloacetate must be converted to either aspartate or malate, and then can be transported to the cytoplasm. This process uses NADH in order to reduce oxaloacetate to form malate. Once in the cytoplasm, malate is again oxidized by malate dehydrogenase to form oxaloacetate using NAD+.
As the next part of the pathway will take place in the cytosol, phosphoenolpyruvate will be hydrated by enolase, phosphopyruvate hydratase, to form 2-phosphoglycerate. Then 2-phosphoglycerate will be isomerized by phosphoglycerate mutase to form 3-phosphoglycerate. Next, with the requirement of ATP and phosphoglycerokinase, 1,3-bisphosphoglycerate will be formed. In the next step, 1,3-bisphosphoglycerate will be transformed to glycerolaldehyde 3-phosphate. This process requires the reduction of glycerolaldehyde 3-phosphate dehydrogenase and the replacement of NADH to NAD+. In the next step, aldolase catalyzes an aldol concentration of DHAP, dihydroxyacetone phosphate, and glycerolaldehyde 3-phosphate to form fructose 1,6-bisphosphate. Then fructose 1,6-bisphosphate is hydrolyzed by fructose 1,6-bisphosphatase. This enzyme can shut down gluconeogenesis at the same time that can activate glycolysis. Next, fructose 6-phosphate will be isomerized to glucose 6-phosphate by the help of phosphoglucose isomerase. As the next step enters the ER membrane, glucose 6-phosphate will be hydrolyzed by glucose 6-phosphatase to form the end product, glucose, giving off an inorganic phosphate. As one glucose molecule is the end product of gluconeogenesis, it will be distributed to the bloodstream by the ER vesicles that contain glucose. The regulation of gluconeogenesis. This cycle is regulated through different ways. 1. Substrate delivery, controlled by what is coming out of muscles, such as alanine and glutamine, and controlled by triglycerides. Another way would be mass action on enzymes, the ratio of NAD and NADH, which regulates the direction of the pathway. Another way would be allosteric activation of key enzymes, such as cyclic AMP. Another, change in mass of enzymes by gene expression. It also changes in amount of enzymes in the liver, PepsiK via cyclic AMP. Fetal cyclin is also prevented by coordinate regulation. This cycle regulates reverse and forward reactions. An example would be fructose 2,6-bisphosphate activates PFK1 and downregulates fructose 1,6-bisphosphatase.
A disease unique to gluconeogenesis is von Gierke's glycogen storage disease. This is caused by an enzyme defect in glucose 6-phosphatase, so that glucose 6-phosphate cannot be converted into free glucose, but is metabolized to lactic acid or incorporated into glycogen. The liver and kidneys are involved, and hypoglycemia is a major feature. Large quantities of glycogen are formed and stored in hepatocytes, renal, and intestinal mucosa cells, causing the liver and kidneys to become enlarged. The condition is inherited as an autosomal recessive disorder. Prevalence is unknown, and the annual incidence is about 1 in 100,000 births. GSD type 1A is the most frequent type, representing about 80% of type 1 GSD patients. Glucose 6-phosphatase deficiency is the cause of type 1A. Presentation is usually soon after birth, but can be a little later. Shortly after birth, hypoglycemia and lactic acidosis often cause convulsions. More moderate hypoglycemia can cause irritability, cyanosis, hypotonia, tremors, loss of consciousness, and apnea. During the first weeks of life, the liver is normal in size, but it enlarges, sometimes very considerably, to cause marked abdominal distension. Growth is retarded and height is usually below the 3rd percentile. Puberty is delayed, but mental development is usually normal. Blood glucose and pH are usually low, with elevated lactate, uric acid, triglyceride, and cholesterol. Renal function tests, creatinine and urea may be raised if renal function is impaired. An ultrasound should be used to assess and monitor the size of the liver and kidneys to detect possible hepatic adenomas and nephrocalcinosis. Glucagon does not cause a rise in glucose levels, but it does raise lactic acid levels. Oral galactose and fructose fail to increase glucose levels, but plasma lactic acid levels increase. Definitive diagnosis involves assessment of glucose 6-phosphatase activity in fresh and frozen liver tissue specimens. Histology shows increased amounts of normal glycogen as well as fatty infiltration of the liver. Kidneys may show glomerular hypertrophy. The main aim of treatment is to correct hypoglycemia and maintain normal glycemia. It is thought that preventing hypoglycemia, which is a particular problem at night, will reduce many complications. Unique aspects of pyruvate carboxylase. Pyruvate carboxylase is activated allosterically by acetyl-CoA. This enzyme is a tetrameric protein carrying 4 molecules of biotin. In animals, the reaction catalyzed by pyruvate carboxylase is the most important anaplerotic reaction, particularly in the liver and kidneys. Pyruvate carboxylase is the only enzyme of gluconeogenesis in the mitochondria. It requires pyruvate and oxaloacetate to be transported across the mitochondrial membrane for gluconeogenesis to occur. Gluconeogenesis' role in homeostasis. Regulation of blood glucose is controlled by the insulin-glucagon ratio. Insulin-glucagon ratio varies from 5.0 to 0.5. Diabetic ketoacidosis can be from 0.1 or less. A low insulin-glucagon ratio will have several metabolic effects. First one could be decrease in glucose transport in muscle and fat. Number 2, inhibit glycogen synthase and activate glycogen phosphorylase. Number 3, stimulate liver gluconeogenesis. Number 4, stimulate breakdown of protein for gluconeogenesis and inhibit protein synthesis. Number 5, inhibit lipogenesis and stimulate fatty acid oxidation. When ratio falls to 1.5, ketone bodies will be formed to spare glucose. This enables the brain and muscles to continue to function and eliminates the need for protein degradation for gluconeogenesis. Creatine, made from arginine and glycine in the kidneys, shuffles between creatine and crenatine phosphate, which both break down to creatinine. Glucocorticoid hormones, cortisol, is necessary for gluconeogenesis and for stimulating release of gluconeogenic precursors from muscle and other tissues.